Hello everyone, my name is Kshiti Patel, and today I am presenting the very important technique. The name is recombinant protein and their use in medicine world. Well, to start with, um, the recombinant protein technology, their main purpose is to synthesize the desired protein through the cloning of the selective DNA. Well, this DNA is called the recombinant DNA, and the technology is determined as recombinant DNA technology, which will produce the recombinant protein, which is our main part of the discussion today. So basically, we will search a gene, will give us the desired protein, and that gene will be taken off, and it's called as recombinant DNA. This recombinant protein is used in a variety of applications like in medicine, in research area, in biotechnology. Recombinant protein used as medication means the therapeutic protein is very popular nowadays to treat popular diseases like cancer, diabetes, hemophilia, and anemia. Well, at research level, it gives the efficient performance in laboratory techniques like Western blot, ELISA, and it plays actually a very vital role in protein-protein interaction as well. It helps in hybrid food production, animal health, and performance at the biotechnology level. Now, the question is, was this technique used in past? Uh, Thinking about this question, yes, but the old technique included the separation of the desired protein through natural source, which was a bit an inefficient and very time-consuming method as well. While in the modern technology, cloning of the specific DNA and the expression of the protein are the main steps to produce the recombinant protein. And the protein expression can be carried away from different types as we can see in figure number 7 here. Over here, the target gene is implanted with the plasmid, and the expression can be either mammalian, insect, yeast, or prokaryotic, and they auto-replicate and they will give us the recombinant protein, which will be the desired protein, um, which was synthesized from our desired DNA. So now, how this technique actually works. The gene of interest, which gives the desired protein, that gene will be taken off from the genome by enzyme restriction nuclease, as we can see in figure 8. This specific gene will be implanted to the plasmid who works as a vector, means the molecular vehicle for that gene, and becomes the recombinant DNA. Well, this recombinant DNA will be inserted into the host cell like Escherichia coli here in figure 9, and these plasmid are usually auto-replicable. They make lots of copies of the same DNA, which will produce the high amount of recombinant protein, followed by the processes of transcription and translation. Now, going a little bit further, from the wide applications of this technology, medicine is very popular for the usage of the recombinant proteins. To take an example here, the insulin is well-known recombinant protein for the treatment in diabetic people. While referring figure 8, pancreas of diabetic patients who are having type 2 diabetes that produces less insulin, which cannot connect to all the molecules of glucose, so the glucose would not be absorbed by the cell wall and the blood glucose level will go high. As we can see from the figure 11, the normal how much amount of insulin can be produced from the normal people's pancreas and from the diabetic people's pancreas. Well, the treatment for this disease that insulin can be produced via recombinant protein technology and will be inserted to the people who are diabetic. Well, as seen in figure 10, insulin producing DNA is connected to the plasmid and then it is implanted to the bacterial cell. Here, the bacterial cell is working as expression of protein, which this bacterial cell will then auto-replicate and produces a lot of insulin, which can be harvested. Well, taking another example of therapeutic protein is generation of antibodies for the cancer therapy. From the first half of the figure 12, the CFH means the complement factor H that secretes anaphylactoxins, which are very important for the immune response of the body. Now, the peripheral B cell produces these antibodies, so they will be cloned and expressed to make the recombinant protein. Here, the CFH antibodies are the recombinant protein, so we are producing these antibodies so we can insert these antibodies to the people who are having cancer. So now these antibodies will attach to the tumor cell and will generate its lysis. So it should be given to the people who are having cancer via any type of form, either medication or injection or yeah. Now, overall this technique is used to synthesize the high quality of recombinant protein. This technology has been used from last almost 30 years, but it has grown a lot in last 10 years due to the innovation and advancement of the biotechnology and the research. Well, it has its limitations as well, but still the usage of this technique in medicine world is so high, so it's not even comparable. From the figure 13, the consumption of recombinant protein in medicine industry via therapeutic use and drug discovery is the highest than any other application. This is the end of my presentation. Thank you everyone for listening me.